In this video, we are going to be covering the concepts of Chapter 3, which is titled Amino Acids and Peptides. When we talk about amino acids, understand that they are going to be the basic unit for peptides and proteins. What this basically means is that peptides and proteins are built from amino acids. In the following figure, we are just representing the different amino acids by the colored circles that you can observe. Understand that when those amino acids are linked to one another, a peptide is formed. Now, the difference between peptides and proteins is just the number of units that we have. And understand that that is going to be addressed in further slides. First, we are going to be going over section 3.1, which discusses how we observe these amino acids in a 3D world. So when we think about the amino acids, the word amino acids is because these basic units for proteins have some familiar functional groups from organic chemistry. So the word amino comes from the amine functionality that is present in the amino acid, and the acid portion of it comes from the carboxylic acid functionality that is present in their structure. Understand that the carbon that is holding all the different components of an amino acid is known as the alpha carbon. And this alpha symbol is going to be the Greek letter that symbolizes that the alpha carbon is the central carbon. Every amino acid contains, attached to that central carbon, in other words, the alpha carbon, a hydrogen atom, a amino group, a carboxylate group, and the R group. So understand that the amino group is the amine functionality that I just mentioned. The carboxylate is going to be the carboxylic acid functionality. It's just that, as we're going to see, changes in pH are going to affect how those functional groups actually look like in an amino acid. Now, when we think about these amino acids, because we have a carbon atom that has 4 different groups, as you can see here in the image that we have to the left of the slide, many of the amino acids, in other words, the majority of them, to be honest, are going to be chiral. Now, understand that when it comes to actually the isomer that is present for them is going to be the L isomer. Now, understand that this is going to be in further slides, but I want to introduce the idea. The only amino acid that is not chiral is going to be glycine, as we're going to observe in a few moments when we go over the structures for them. Now, what is the difference between the amino acids? These R groups. And R, remember, that is just an abbreviation for the rest of the molecule. So, each one of the amino acids is different from one another because of the R group that they contain. Now, Because amino acids have an amino side and a carboxylic acid side, as you can see, they can act as acids or bases. So that means that amino acids, as we introduced the term in the previous chapter, are amphoteric. Let's look more at the chirality of amino acids. So, as I mentioned in the previous slide, most of the amino acids that we observe in proteins are going to be the L isomer. So, the L and D configuration that is utilized in biochemistry to symbolize the chirality of some of our biomolecules, understand that comes from the idea that L stands for levus, which means left, and D stands for dexter, which means right. So understand that this is very related to the concepts of R and S configuration that you have learned in organic chemistry. So if we think about it, previously you saw that things that were towards the left, okay? And if we think about the situation that we have here, when we were prioritizing RNS configuration, remember that we do it based on the atomic number of the atom. So I'm just going to utilize it for specifically the L isomer, so you can see how this relates to RNS configuration. But understand that in biochemistry, we are always going to talk about L and D isomers. So from your background in organic chemistry, you understand that whenever you have a carbon that is chiral, we can actually determine if the configuration around that carbon atom is going to be R or S. The priority when we're determining R and S configuration actually comes from atomic number. So we know that when we look around, here we have hydrogen, we have nitrogen, we have a carbon atom bonded to the central carbon, and the R group, for the most part, that we have in many of the amino acids is always going to be in carbon atom. So when we do the priority, understand that the nitrogen is going to get priority 1 because it has a larger atomic number compared to carbon. Priority number 2 is going to be the carboxylate group because it's attached to oxygen compared to the side chain, which is going to be priority 3. And then the priority 4 is going to be the hydrogen because it has the smallest atomic number. So if we relate the D and L configuration of amino acids to RNS configuration, you can see that the L isomer, because our hydrogen is already pointing towards the back, this specifically is going to be counterclockwise. So the L isomer is actually the S configuration. And then the D isomer is going to be the R configuration. But again, this is just to relate it to previous chemistry courses. Understand that for the rest of the semester and throughout this course, we are going to refer to the chiral centers in the biomolecules in which we focus on chirality as L or D isomers. Now, understand that these amino acids are going to be grouped based on the characteristics of their side chains. So, the major 4 groups for amino acids are the nonpolar amino acids, which they can be aliphatic or aromatic. Aliphatic is a term that is utilized when you have straight chains of carbons and hydrogens. We're going to have some amino acids that are polar, and sometimes they are categorized as polar neutral. We're going to have amino acids that are acidic, followed by the amino acids that are basic. Understand that these characteristics, as is mentioned here, is based on the side chains, because remember that every single amino acid has an amino group and a carboxylate group. So these categories are based on the side chains that are present in them. Let's look at them more closely in terms of their structure. First, let's talk about the nonpolar aliphatic amino acids. As I mentioned, an aliphatic is going to be where we have a straight chain of carbons and hydrogens. So when it comes to the amino acids, I'm just going to write a note here. You must know their structure, condensed or skeletal, their full name, their one letter code, and their three letter code. And you will see that this is going to be present in the sequential slides that we have in this chapter. So again, let's go back to talk about the nonpolar aliphatic amino acids. So one thing that you can notice here is that the amino acids that are part of this group include glycine, alanine, valine, leucine, isoleucine, methionine, and proline. One of the things is that it is important that you guys also know the functional groups. Present in the side chains of the amino acids. So one thing that you can notice here is that glycine, alanine, valine, well, mainly alanine, valine, leucine, isoleucine are going to have alkyl groups as a side chain. Because glycine, as I mentioned before, is the only amino acid that is not chiral, because as you can see, it has two identical groups bonded to the alpha carbon. So alanine, valine, leucine, isoleucine will have an alkyl group. So an alkane, in a way, chain as its side chain. Now, methionine has a functional group that is not typically talked about when it comes to organic chemistry. And this is going to be a thioether. And the reason why it's a thioether is because, as you can see, we have a sulfur in between carbon atoms. So that's the ether portion. And then the thio comes from the sulfur atom itself. Now, as part of the nonpolar aliphatic amino acids, understand that we also have proline. Proline is going to be one of the only amino acids out of the crew that the R group is bonded to the amino group within the amino acid. So I'm just going to highlight it to illustrate it. As you can see, the R group, this yellow part, is actually bonded even to the amino group in it. Technically, that group is not called aliphatic because it's actually cyclic. So proline is specifically known as an alicyclic amino acid. Let's continue talking about the amino acids. So out of the nonpolar amino acids, we also have amino acids that are aromatic. And as you can see, as part of that, we have phenylalanine and tryptophan. Phenylalanine specifically has a benzene ring, so it has an arene group. And one of the things is that when it comes to tryptophan, and we observe its side chain, which includes the CH2, specifically at the side chain, this is going to be composed of an arene, and even though it has a heterocyclic amine, the lone pair in that nitrogen is not involved in any basicity. It's actually involved in aromaticity. But this heterocyclic amine, its lone pair, is not accepting a proton because it is part of the aromatic system. The next group of amino acids that we have are the polar, or in other words, the polar neutral amino acids. And as you can see here, we specifically have at least 3 amino acids that contain a hydroxyl group. Serine, threonine, and tyrosine contain a hydroxyl group. Cysteine contains a thiol group. And as we look around, as you can see, the major functional group in the side chain for asparagine and also for glutamine is going to be an amide bond. When it comes to the acidic amino acids, understand that aspartic acid and glutamic acid in the side chain are going to have a carboxylate group or a carboxylic acid. So they are acidic amino acids because of the carboxylic acid slash carboxylate group in the side chain. So again, the two members of that, or the two amino acids that are part of the acidic amino acids, is going to be aspartic acid, which if you have a carboxylate, it is also known as aspartate. And when it comes to glutamic acid, when you have the carboxylate, it is known as glutamate. So understand that those names can be used interchangeably. From the last group of amino acids, as you can see, we have the basic amino acids, and there are 3 members of this family. We have histidine, lysine, and arginine. So understand, when it comes to this, histidine is going to be a heterocyclic amine but understand that different from what we observe in tryptophan, specifically one of the nitrogen atoms, the one that I circle, actually has basicity to it. That's why we see it protonated. Lysine is going to have an amino group at its end, which is the one that acts as a base. And when it comes to arginine, the following group that we see here, which is a carbon double bonded to an NH, single bonded to an NH2, single bonded to an NH, and then the rest of the side chain, that is known as a guanidinium group. This guanidinium group specifically at the nitrogen that is double bonded to the carbon is where the basic nitrogen out of the system actually is present. Now, When it comes to amino acids, I already explained that some of them we have to treat them as special just because of the features that they have. So remember, protein, when it comes to proline, it is again a secondary amine, but because specifically the side chain is bound to the amino group at the alpha carbon. That's why it's going to be a secondary amine. Remember that it is a cyclic amino acid. Glycine is the only one that is achiral. Cysteine is the only one that we're going to see in the future, how it actually has bonds that can be oxidized and they form disulfide bonds. And understand that amino acids are important to be obtained through the diet, but there are a number of amino acids that cannot be synthesized in the body, so we must obtain them from our diet. So those are known as essential amino acids. They include histidine, leucine, isoleucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Now, we always talk about that there are 20 amino acids, 20 amino acids, 20 amino acids. Actually, the number of amino acids that are known to be found in the human body are more than 20. Now, because the extra amino acids are not found in every tissue and in every cell, that's why we refer to the primary 20. And these that you can see are considered uncommon amino acids. So, understand that these uncommon amino acids are going to be produced by post-translational modifications. So what this means is that after the protein has been made, the modifications are going to be happening at the amino acids to give rise to these uncommon amino acids. So to talk about the ones that are been observed so far, we have hydroxyproline. And as you can see, the difference between proline and hydroxyproline is that specifically when we look at the ring system, I'm just going to highlight it so you guys have noticed what's the difference. In hydroxyproline, at the second carbon from the side chain, we have a hydroxyl group. When it comes to hydroxylysine, as you can see, that hydroxy group is going to be added on the second carbon that is going towards the alpha carbon away from the amino group on lysine. Now, those two uncommon amino acids, hydroxyproline and hydroxylysine, are actually found in connective tissue. For example, we have observed them in collagen. The last uncommon amino acid that is known to be present, as you can see, is tyrosine. And then in tyrosine, it is actually a derivative of tyrosine. So tyrosine, what happens is that it is produced only in the thyroid gland. And when we look at the difference in terms of structure, you can see that we have iodines that have been added to the benzene ring that was part of tyrosine, and in addition we have an extra group that is another benzene ring that has two iodine groups and a hydroxyl group. So again, these are examples of some uncommon amino acids that are still present in the human body. So when it comes to amino acids, what exactly should you know about it other than the information that has already been presented to you? Well, let's actually do some practice. So when we look at these amino acids, as you can see on problem 4 in the textbook, it states, identify the types of amino acids in the following peptide. Problem 4 in the textbook actually wants to know that or which one of those amino acids are polar or aromatic or sulfur containing. Okay, so I'm just going to classify all of them because it's just good practice that we classify all of them. So let's just go through them one by one. So if we review the previous slides, understand that these amino acids have a classification. So when it comes to this, we start with valine. Valine, as you may remember, is going to be part of the nonpolar aliphatic amino acids. I'm just going to use little abbreviations for the categories. So I'm going to use NP for nonpolar. I'm going to use P for polar. I'm going to use A for acidic. And I'm going to use B for basic because it will make it easier. Now, I'm just going to generally classify the amino acids that we have in the following peptide. As you can see, first we have valine. Valine is going to be one of our nonpolar aliphatic amino acids. We have methionine, which is also part of the nonpolar aliphatics. We have serine, which is part of the polar amino acids. We have isoleucine, which is nonpolar. We have phenylalanine, which is also going to be nonpolar. We have arginine, and if you remember, when it comes to arginine, it's going to be part specifically of the basic amino acids. We have cysteine. Cysteine is going to be specifically out of the polar amino acids. We're going to have tyrosine. Tyrosine is also a polar amino acid. And lastly, we have leucine. Leucine is a non-polar amino acid. So in this little peptide, we do not have any of the acidic amino acids. And remember that that is glutamic acid and aspartic acid. This is the 3-letter codes for the amino acids. Now, taking that into consideration, understand that then we can actually say, okay, so taking that into consideration, we have to think, okay, which ones are aromatic, okay? And in essence, understand that they are going to contain a benzene ring in their side chain. When it comes to this, and let me just use highlighter tool. Out of my polar, as you can see, I have serine, I have cysteine, I have tyrosine. Now, based on my highlighter tool, when it comes to the ones that are aromatic, if you remember, okay, phenylalanine contains a benzene ring. Out of the rest of them, tyrosine also contains a benzene ring. Okay? Remember that the other one that contains a benzene ring is going to be tryptophan. When it comes to the system. But as you can see, we don't have tryptophan here. Then, out of the sulfur containing, and I'm just going to use pink for my sulfur containing, there's pretty much 2 amino acids that contain sulfur. One is methionine because it has the thioether. The other one is cysteine because it has a thiol group. So pretty much if you're given a peptide, you can identify specifically what kind of groups you have present in the molecule. Now, there is another type of problem that you guys have to be able to identify out of the whole system. And that is, if you are giving a peptide, can you identify with the one-letter codes the amino acids that are present in a given peptide? As you can see here, we have in these structures the skeletal structure for the amino acids. These peptides have been drawn at a particular pH, but understand that that is going to be discussed later in this chapter. So when you have a problem like this, the important thing that you have to remember is that in sequences of amino acids in a peptide, there is a trend, okay? They are always organized in the same way. So when it comes to this, understand that a peptide, it is always drawn from the amino side, ending always in the carboxylate side, okay? So the pattern that exists here is that we always have, and I'm going to use my highlighter tool to illustrate this. We have nitrogen, alpha carbon, C double bond O. 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 And as you can see here, we have the carboxylate. So understanding that pattern tells me that when I'm looking for what amino acids do I have here, remember that out of the alpha carbon, I have the side chain stretching from it. So the best way to approach this problem is to actually take note or highlight where my alpha carbon is. So as you can see, I have alpha carbon, C double bond O, N, alpha carbon, C double bond N, alpha carbon, C double bond O, N, alpha carbon. And then I have the carboxylate. So what does that mean? That in the 5 highlighted alpha carbons, that's where my side chain is. And based on the side chain that it is there, I can determine the amino acid that is present. So let's actually categorize each of them. So in the first one, I have a side chain that is connected to the amino group. So that means that I have proline present, and the one-letter code for proline is P. If I look at this next one, the next amino acid that I have is going to be a methyl group extending. Methyl group is going to be alanine, and the one-letter code for alanine is an A. Then I have CH2, 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 and I have this guanidinium group, which means that I have arginine, and the one-letter code for arginine is R. In the next one, I have CH2, 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 NH3, which this is lysine. And the one-letter code for lysine is K. So the first peptide is PARK. Let's move on to the second example. Okay, this is one of a little bit of a longer one. I go through the same process. Let me find the pattern. Okay, I have an amino group here. I have the carboxylate. Remember, the amino is the beginning, the carboxylate is the end. So I have nitrogen, alpha carbon, C double bond O. 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 Nitrogen, let me just erase that. Alpha carbon, C double bond O. Nitrogen, alpha carbon, and then I have the carboxylate. So that means that, as you can see here, this is a longer peptide. I have 1, 2, 3, 4, 5, 6, 7, 8 amino acids here. So I'm going to be using my highlighter tool to show these. So the first one that I have is CH2OH. So that is serine. So serine is a capital S. Next amino acid that I have is a methyl group. The amino acid that has a side chain that it is a methyl group is alanine. So the letter A is going to be the one letter code. Next amino acid, I have CH2CH2 and it's binding to the nitrogen. So this is proline, which the one letter code is a P. The next one I have CH2CH2O and it's binding to the nitrogen. That is another proline, which is a P as a one letter code. Next amino acid, I have a CH2, okay, with this heterocyclic amine. This is histidine, which H is going to be the one letter code. Here I have, as you can see, a sec-butyl group. And the sec-butyl group is actually for isoleucine, which I is going to be the one letter code. Here I have a side chain that it is CH2CH2CH2 and then the guanidinium group. That is arginine, which the one letter code is an R. And lastly, I have CH2CH2 C double bond O, O minus, which that is going to be glutamic acid, which the one letter code for it is the letter E. So the second peptide, as you can see, these are going to be the one letter codes for their amino acids. Make sure that you utilize the resources in the textbook, so the problems at the end of the chapter, or if you find other peptides, be able to do this problem. There is a question about this in the chapter quiz for this section of the book.